Hi everyone, in today's video we're mainly going to talk about proteins and the chemistry behind them and their amino acids that they're made of that kind of dictates how they fold. And that's one of the major mysteries of biology is how proteins fold into the unique 3D structures that they end up being, because there are so many possibilities of that 3D structure. It gets really interesting. To start off, I did want to just kind of review with some concepts and apply some concepts to what we saw with DNA chemistry. And we're actually going to turn first to an enzyme. And so we saw these when we were kind of talking about bacteria, and these are restriction enzymes. These are enzymes that cut regions of DNA at specific sequences, and these evolved in the bacteria because, for example, the EcoR1 enzyme evolved in bacteria that was fighting against a virus that had a sequence similar to the one here in orange, but this sequence was not present in the bacteria itself. So this could basically snip the virus like this. We use these as tools today to cut DNA at these regions so that we can reattach them to new pieces of DNA that have these matching ends, like right here. So this is really good, and it's a good example of where we're going with enzymes. So typically when you think of enzymes from a Bio 101 perspective, it's that they facilitate chemical reactions. We will see in this class the number of enzymes that it takes for genomics. They do wild things. This is the kind of the beginning of that, where you're going to start to see that enzymes are capable much more than just shifting chemicals around, okay? They're capable of breaking things right at the right spot and then reannealing them or something like that. So keep that in mind, as the idea of an enzyme should be something very fluid and very large when we're going into this kind of biology. More on this application example though, this is a very good way to not only measure what you've cut, but agarose gels basically form these little saccharides that inhibit large pieces of DNA from going through this gel right here very quickly. So if you're large, like right here, it basically takes more time to wind through the polysaccharides, the agarose that the gel is made of, than if you are, let's say over here, number 2 is a smaller piece of DNA. This can kind of go through really quick because it's so small. The reason it ends up like a line is that you load it in a well up here, and then you turn on the electrode. And remember, DNA is negative, so it's going to push this way. All the DNA is going to push this way, out this way. This is also really good for getting rid of protein because the proteins don't travel through the agarose gel very easily, so you only visualize the DNA. So we'll cover more on this when we cover anything in lab related. What we are going to cover is a pretty quick lab, where we actually use a few restriction enzymes to identify sequences of DNA. And so this is a good example of, actual enzymes that people use in lab to cut at certain regions. You can see that some of these cut regions that have an N. There is no N nucleotide. What N signifies is that this could be any of the nucleotides, right? Some, you'll notice too, don't leave sticky ends. These are called blunt cutters, basically. These are going to be double strand breaks right here that actually don't have a sticky end. These can still be very useful for research, but it's something that when you have to reconnect them is called blunt end cloning, because these regions aren't actually going to stick the same way. See how this one is going to have these two sticky ends, this one won't. Okay, this is also good review, and I kind of wish this slide was in the stacking that we talked about in the last video. So remember that this phosphate backbone right here is super negative, so all the water around it is going to be happy to kind of saturate this backbone. These bases don't have much charge to them. They're pretty nonpolar and they are hydrophobic, so thus they're going to stay kind of within the other hydrophobic regions. So with DNA, you have nucleotides coming in on this side and binding them and shield them in that structure. We'll see later in chapter 6 with RNA that RNA doesn't have that luxury, and that's another reason why RNA is so unstable, is that these bases are exposed, and what they often do is fold into a hydrophobic core away from the water together. And you can see like how this is not going to happen in nature because there's too much water out here for this base, and it's basically going to hide in here like we see over in the favored. Okay. I know that I should have put this in the last one too, but this is again, a very good overview slide of the interactions that we are going to see here, where you have, these phosphate backbones going back and forth between each nucleotide, and the bases are interacting with two H bonds between A and T, and 3 H bonds between G and C, making G and Cs actually a little bit stronger than the A and Ts. So a sequence where you have more GCs, that's actually a tighter sequence of DNA. So we'll review that again when we get to the wild DNA structures in Chapter 5. I think right now we're calling this Chapter 4 for proteins, but this is just a good review here. And again, yeah, purines pair with pyrimidines because you need a double with a single right here. You need a double with a single right here too. Otherwise, bumps are going to form. If you pair two, like a guanine with an adenine, a big like gash is going to form in the DNA, and that's going to, you know, alert your body that there's or your cells that they're bad. Okay, so we are at proteins, and like I said before, proteins are the key to the functions of a cell. They are the key to a functioning organism. When you have a damaged protein, you will possibly incur damage to your cells. You can damage DNA like we saw and have silent mutations and it won't do anything, you know. But if you damage a protein or you lose it, that's when you start seeing something bad. The other thing is, can we create something, for example, that can do anything? Most of the biotechnology that we have, even a protein that's advanced as CRISPR, we don't know how to predict to create something like that. We borrowed CRISPR from bacteria. We borrow restriction enzymes from bacteria. We borrow green fluorescent protein from jellyfish, okay? Most biotechnology is stuff that we've stolen from organisms. So can we create something for use that we specifically designate? And the answer is that we are not capable of that yet. We are not capable of predicting a 3D shape that could perform these structures, for example. We don't know how to design a perfect enzyme, because in this nice cartoon like biologists like to draw, there are, you know, thousands of interactions and foldings and all kinds of stuff that make this, you know, right here the active site or something. To form a linear stretch of amino acids that fold into this, where all these amino acids are interacting, they have to interact with hydrophobic and then hydrophilic regions. We, in all our technology and all our machine learning, all our AI, we cannot predict that. We do not know how to create that yet. Our best usual bet is that we try and create antibodies that fight disease, basically just by training our own immune system to do that. This is what we do with vaccines. We send in something that will cause an antibody response that makes new antibodies to whatever we're sending in that will bind to these variable regions and destroy it. But the problem is that you basically can't do that de novo. We still need to rely on the body to figure that out. So again, as important as DNA is, and as dynamic and cool as RNA is, it is the single strands of polypeptides that eventually do function in the organism. And this is kind of what I was referencing when, you know, this is kind of your Bio 101 look at what enzymes do, is that there are chemical transitions that need to be made that require energy. But if you have a shape that can facilitate this interaction, so this active site in our little enzyme Pac-Man here, this can facilitate a otherwise random reaction to happen in here, because you're going to have amino acids that are going to grab whatever your reactants are and put them in a position that will turn them into products. Now, again, like I said, this is much bigger than just, you know, digesting things or something like that. This is adding nucleotides. This is editing DNA. This is scanning DNA. So any enzyme function is likely something having to do, you know, with that perfect active site. And it doesn't always have to be like a clean chemical equation. You'll see that you can make it that, but for me, it's easier to visualize that it's not. So we already covered this again, but it's good to re-up that these are very complex things. And like we talked about, these R groups are going to be interacting with each other. Some are hydrophilic, some are hydrophobic. Some like binding to each other, others don't. So we are going to get into some very dynamic stuff. Like we talked about too, it's not necessarily the DNA that is going to determine the phenotype. If that makes sense? And as you can tell here between these three adorable species, especially right here, the DNA between them is really, really similar, okay? The 10% is actually kind of a long shot. It's actually probably even more similar than that, but you can see the diversity in size, makeup, you know, different senses, and this all comes down to, again, epigenetics that control the amount, you know, what proteins are on and off and when. So proteins themselves are what really determine what an organism looks like, because you're going to have different cells that produce different combinations of genes. Even if they have very similar genes between them, it's a big combination of what proteins are actually going to be different. Now, there are differences in the actual conserved proteins between these three. It's just less than, you would imagine, given their diversity of form. Okay, so again, we saw this here, where yes, DNA is the blueprint, and you can have this nice normal protein, but if you ruin your DNA blueprint, yes, you can ruin the protein and or completely lose it. So in this case, I know we talked about this. This is the sickle cell trait, and all this is a missense mutation at this 6th position in this linear sequence of amino acids. So we just changed one thing, and this is a glutamic acid, so I mean it's a big charged molecule, and you go from that to a valine, which is pretty small and not charged. So what that does is that you have this B subunit 3D shape, and you have this B subunit 3D shape, and the key is that you have an exposed hydrophobic region all of a sudden in this 3D shape. What that is going to do is cause this interlocking mechanism to happen. See right here, these hemoglobins, in this case, the gene that you've basically changed up here, they go in these little nice single units. Here they link up because you have an exposed hydrophobic region. And this is a key tenet of how proteins and their 3D shape form. If you have basically an exposed hydrophobic region like this, it's going to find the other hydrophobic regions and bind up with them. It's not going to face out freely like it should be right here. And you end up with these long striated structures that cause the sickle cell trait. Now remember, this means that if you're heterozygous for this, you got half of these and half of these, so O2 flows pretty well, and these cells prevent enough of these for allowing malaria to basically hitch a ride. So again, being immune to the top disease of humans is not something I'd necessarily call a disease. So I think it's much more advantageous than people usually give it credit for. How proteins denature. So this kind of highlights the importance of the 3D structure. When you have a normal protein, it is in a 3D shape, as you can see here, where things are interacting at the right spots, and maybe you have active sites somewhere that are in the right position. Everything's fine. If you denature it, whether that's through pH differences, heat, anything, most proteins come out as, like you can tell, this long sequence of amino acids. This is the sequence that even human scientists like us, we don't know how to create a linear sequence and a combination of amino acids that go towards a specific function. We're still not there yet. The other problem with denaturation is that sometimes you can return the protein to its natural environment and maybe it'll renature. There's no guarantee of that though. So sometimes if you undergo like a very distinct or bad environmental condition, the proteins don't go back to their good shape, right? So despite that, another major cause of denaturation is, let's say one of these, core proteins is messed up here. This could cause the entire thing not to bind at the right spot, and thus the whole thing basically denatures. So that's why even small mutations can have a profound difference. Okay, so we've all seen this before, but these structures and the way protein basically manifests that 3D shape and structure is very important, and it's all written in primary structure language. So, I mean, here's the challenge if you want to make synthetic proteins that can do anything. You have to make this structure, and you just do it in a straight linear line. You're going to have to predict how this straight structure of amino acids could possibly form specific secondary structures. You're going to have to predict how those secondary structures, and look how many of them there are. We're highlighting this nice purple one, but look at all these alpha helices in this little active site here, and they're surrounded by these beta sheets, beta turns in here. You have to be predicting the entire time how many of these secondary structures are going to interact to form the full tertiary structure. And if you really want to get complex, you're going to have to create multiple proteins, as you see here. And that's what quaternary structure is. It's either isoforms or different genes binding up to make a single functional protein. So in each of these cases, the complexity of the chemical interactions of these diverse amino acids, it's massive. It's so massive that we have no way to just create a sequence and say, yes, this protein's going to do this. We have to rely on what nature has evolved and given us. So let's bring up a clinical scenario and something fun with the immune system, which I think is the great unifier of biology is immunology. Your immune system is trained from a very young age. It is educated what yourself and your cells look like. Okay? It is trained to detect what looks like you and what doesn't. Specifically, it knows that viruses just adventuring around, these are nasty looking. Remember, they were like, like a big hexagon with DNA in them. Oh, nice. And they're running around and they're trying to insert in cells. These are really foreign looking. You have a lot of cells that just gobble these up. Bacteria, remember, these are prokaryotes, so they look really different than your eukaryotic cells. Fungi, again, they're eukaryotes now, but they're pretty different. Your immune system's pretty good at sniffing out fungi. You have a lot of innate antifungal parts of your immune system. Large parasites caused us trouble for a long time. Same with things like malaria, protozoa. So these are eukaryotic cells that in some cases actually feed on your cells too. This is when, virulence isn't necessarily, over here virulence isn't a bad thing. You might just be a good home for viruses or bacteria or fungi. These are legitimately basically animals trying to kill you. This is much less like, oh, you're just a good environment. No, you're kind of the prey when it comes to these two things. But they look pretty similar to your eukaryotic cells, so they're a little more challenging. This is where these three are where things start to get a lot harder. A virally infected cell is still one of your cells. It just starts to behave and look a little different. It might express a few different surface receptors, things like that. Cancerous cells, these are really hard. Okay? The only difference between a healthy cell and a cancerous cell is maybe a few different mutations, maybe some genes are on when they should be off, things like that. Now, the cool thing is that your immune system is hypothesized to actually be able to hunt down cancerous cells before they become big tumors. And they can do this by just detecting even the slightest differences in protein expression that should not be there on the surface of these cells, and they decide to basically kill them. Your immune system is a very powerful thing. And that brings us here to prions, also a very good example of protein biology. All a prion is a misfolded 3D shape of one of your brain proteins. I'm not exactly sure what this normal prion protein does in the normal brain. We think it has something to do with copper signaling of some kind in your brain chemistry. In any ways, it's pretty, important for whatever reason. But all a prion is one of these proteins that has been misfolded in its 3D shape. Its primary sequence is the same thing, okay? But for whatever reason, its tertiary structure has changed. And its tertiary structure has changed into something that has an active site that folds more of itself into the bad prion form. It's basically like a zombie, okay? Prions find other prion-like proteins, fold them, and make them into a 3D shaped prion, all without changing or mutating anything. So poor mad cows. This podcast will kill you does a great episode called Apocalypse Cow on the history and discovery of prions. It's actually very, very, very good if you have time to listen to that. And this is kind of just the basic sequence of what a prion really is. So in green, you have this normal prion precursor protein that humans and a lot of other mammals kind of have in their brain. If it misfolds, for whatever reason, now you have this active site here. And this active site exists to go find your normal proteins, misfold them into a new 3D shape, so basically force a chemical reaction like enzymes do, and now you have two of these. Okay, like I said, it's basically like a zombie. When it comes in contact, it's going to force that change. The unfortunate thing for our immune system is we have very little ways to detect this. Because this is not mutated, it looks a little different, but it is a human protein. What happens is that these prions don't get degraded the same way that the precursor usual normal one does. So these are different Prion diseases, CJD, Crutchfield's, Jacob's disease, I probably butchered that. Kuru is another one. This is mad cow, and this is scrapie. And this has actually transferred into American deer, and that is what chronic wasting disease is, so maybe think twice about eating venison next time. Now, I have up here cure prions because they hurt the mammal brain similar to the way Alzheimer tau proteins are hypothesized to, is that tau and prions, they don't go away. These dark areas are areas where the prion is accumulating, and it basically floods the brain and doesn't allow connections to happen, okay? So how do we cure something like that? What if we could create a refolding protein and send it into the brain that would come in and refold the protein back, refold the prion back into something else, right? What if we had something like that? The problem is that we have very few ways of understanding the path that a linear sequence takes that ends up at the goal. So even if we wanted to make something that could refold proteins and get rid of prions, or it could get rid of tau proteins, we don't have the capability to predict how that's going to do that. And what these arrows symbolize is something what we'll see in a sec here, the molten globule idea, which is as energy and energy dictates and entropy dictates, the 3D shape of the protein will form secondary structures that will start to interact with each other and eventually form this big tertiary structure that will all have to depend on the interactions, but we don't know the paths that it will go to do that. We don't know the energetically favorable way that that will come out. So when you think about all the things that we could accomplish, and after this class, maybe you'll even have more ideas, all the things we could accomplish if we could master this, that's when you start getting into some serious jumps in human biology, technology, and medicine. A couple of my students had a great idea. Yeah, they were basically like, I would make a prion. I asked them, how would you cure prions? And I was like, and some of them said, I would make a prion that refolded the bad prions into something inert. I was like, that's actually a great idea. If we could master this, this would be great. All right. These are the two main theories of how protein 3D shape comes to be. Okay. Actually start down here at option 2 at hierarchical, because this is kind of the more straightforward but less likely method in my mind. Basically, all it's saying is that proteins fold basically in the same structures that we have dictated them to exist in. You start with a primary structure. Those all fold into nice, neat secondary structures that are predicted by the chemical composition of the primary. These form tertiary structures. See how there's tons of these secondary like helicases and beta sheets. And then these go and fit with other proteins that have undergone a similar, pretty clean and easy quaternary structure. What the globule model basically dictates is that the protein is going to start being translated amino acid by amino acid. So as it's getting spit out by the ribosome, it starts folding on itself in the reactions that are present for it. And essentially it goes through these transition states called molten globules where, and this does a good job up here, these are the molten globules, this stage right here where it's like this wild, crazy catastrophe of chemical reactions that are happening that eventually settle in. See how those eventually settled in to a lower energy state, because eventually you want to basically minimize energy, but you have to get through all these various immediate states. So what this wide basket here is all the possible states of an amino acid chain. Eventually, as more of it comes out, there are more energetically favorable intermediates here, and eventually you reach just one state it's the very best 3D structure. So that's how complex this question is, unfortunately, and how far we are from addressing how to make something that will do what we tell it to do. Some of the ways we do know how things fold is that as this primary sequence comes out, you have polar and you have nonpolar. So remember, polar like interacting with water, they're hydrophilic. Nonpolar, they don't like water, they're hydrophobic. The nonpolar ones and the hydrophobics, they're going to form a core. Because remember, everything in life, for the most part, is going to be in this nice aqueous environment. See how the polar ones are going to form these hydrogen bonds on the outside, but the inner core of these greens, they're going to just contact each other. This is a similar principle to the hydrophobic core forming with nucleic acids. How DNA interacts with itself, and RNA, when it turns 3D like this, also does something similar, and we'll see that. So this is one of our best ways of actually predicting how proteins fold, is that we have to assume, this is one of our rules that we can assume as we do try to create this, is that something like leucine is going to form a nice hydrophobic core where they're just interacting with each other, because they're hydrophobic too, they're nice and nonpolar. They don't want to react with all the water that's out here, they want to react in here. Oh, and yes, this also is going to dictate, if we want to create something, regions that will interact in the plasma membrane as well. So here are two of the main structures that we've seen before. You've probably seen in your other intermediate classes. alpha helix and beta sheets. Beta sheets are much more rigid, and they have, these nice hydrogen bond structures that keep them nice, kind of planar almost, versus the alpha helices. These are when the amino acids go basically in a pattern that facilitates hydrogen bonds at these lengths right here, right? So we know from all our computing power and our ability to visualize things that structures like this are basically what we can call common and characterized. We can predict the composition of, of a primary sequence, and we're like, that looks like it's going to be an alpha helix, and that looks like it's going to be a beta sheet. The third structure that we are pretty confident in is a beta turn. These are amino acids right here, and they're basically, say the protein is going this way, and it abruptly turns, it's an abrupt 180, basically. And we can predict these because usually what you have in a beta turn is a proline, and these are super rigid because they have this, this 5-piece right here that can't be changed, and it's basically a kink, and then a glycine, which its R group is tiny, it's just a hydrogen, so it's really easy to fit. That hydrogen doesn't take up much room in this big 180 turn, so that's why we see those two very common in a beta turn. So again, remember, if you're trying to create a 3D protein structure, you have to account for the sequence of all these that you're going to spit out and how they're all going to interact, not just in the final state, but in that globule intermediate state. So it's very, very difficult to say the least. Okay, so when we look at those three structures we just looked at, the beta turn, the beta strand, and the alpha helix, let's take a look over here. Proline and glycine. So in this case, in these beta turns, these abrupt turns, you need prolines to be that rigid curve. You need glycines to allow that curvature to come around. Notice that some of the bigger ones don't actually show up in this beta turn. Beta strand is a little different. You can tell that there are some, like these isoleucine and valines, kind of like plain, nice ones. They're found in these, and we can kind of predict that if there's a lot of these ones, it might be a beta strand. And then with an alpha helix, this is actually the one that's designated here from highest to lowest. You can tell based on these amino acids, if there's a lot of these amino acids in a given primary sequence, it's likely forming an alpha helix, and it's less likely if these ones are around. So relative frequency, yeah. So these are how we can kind of predict what secondary structure is going to happen. But then remember, for tertiary structure, we need to remember how many alpha helices are interacting with this beta strand? How many are interacting with this next beta turn? They're all interacting with each other to form that 3D shape. And so like this kind of says, we can predict some of these, some of these structures. These are some other structures, so like a clean out fragment, crystalline domains, that we can kind of predict how they're going to look. But these are still really wild structures that we can't mimic de novo. Again, we can use CRISPR or something to change the DNA pattern and thus change a few amino acids and see the changes. It's very hard for us to design something, completely specific. And because this 3D shape is meant to be this perfect amount of amino acids, oh no, that are going to fit. And usually what you do have for any enzyme or protein that has a function, you do need an active site for that ligand, right? And so any disturbance in the 3D shape, even around the active site, and God forbid the actual active site amino acids, you can completely derail this process of being able to bind to your ligand and actually facilitate the chemical reaction. And remember, in the case of what we're looking at, the ligand is going to be incoming DNA nucleotides or something that you're adding during DNA replication. It's not always going to be this simple, one thing goes in, two chemicals come out. And this is the detailed look in an active site. Okay, you have very specific amino acids interacting with very specific spots on the ligand, okay? And it's a perfect fit because you don't want something going into this active site that isn't a perfect fit, right? So in this case, you can actually tell our ligand is DNA. And this is actually, this active site right here is actually, basically, I think DNA polymerase in this case. So one way that we attack proteins that we do not want on is we can design small organic molecules to plug the active site. We plug the active site by mimicking the exact spots where these hydrogen bonds form. See how hydrogen bonds are forming? But in the case of our targeted small molecules, these will form covalent bonds. That's what ibrutinib a popular lymphoma therapy does. It goes in and binds to this Bruton's tyrosine kinase that is a big like oncogene basically. It drives a lot of growth pathways. But if you plug it with ibrutinib here, these bonds right here don't just form hydrogen bonds in the active site, they form covalent bonds, and they basically kill that protein because they're plugged its active site forever. Because remember, those covalent bonds are super strong. Another cool set of proteins, they're not always degrading stuff. Some are single-strand binding proteins, and they just bind DNA in a nice like wind-up to basically keep it safe during replication, for example. Because remember, when you expose DNA to replicate it or transcribe it, single-stranded DNA could get like attacked by other enzymes. So these binding proteins here actually keep it safe. So again, here this is a DNA binding protein actually doing the interacting. You can see it's really cool. It's only made of a few helices and then one helix that spits out. And the helix is perfectly designed to interact with this groove that DNA forms in the helix. So it does kind of have an active site, or at least a binding site, right? And the cool thing is that a lot of these residues, a lot of the amino acid residues that we have are perfectly built to interact with specific nucleotides. Isn't that awesome? So in this case, specific nucleotides, or sorry, amino acids, you'll often see will have, sorry, that's scalp barking. You'll often see that specific amino acids can bind specific nucleotides very well. Our proteins harness this, and this is why you actually have regions of DNA that are bound by specific regions of protein, like repressors. Remember we talked about repressors turning genes on and off? This is how they actually do it. What these are inverted repeats. See how this light orange is the same down here, but just in reverse? What this lac repressor has right here in these regions are binding sites that perfectly fit the DNA. See, they perfectly fit these regions right here, and they basically like twist it up. See how they grab here and here? They will twist up the DNA because they bind here and there. And the promoter, which we need RNA polymerase to attach to, is no longer available, right? We basically X that out. So this is how, kind of where we're going with how proteins interact with nucleic acids. It's a very specific chemical attraction and reaction. This is another very, key step when we are adding nucleotides. So in the case of, any polymerase, this could be RNA polymerase or DNA polymerase in this case, I think. What you have is this active site right here. For example, you have a glutamine right here and here. The cool thing is actually in this active site for DNA replication, when see how what you're doing right here is you're adding this phosphate chain and you're reacting it. Well, sorry, you're reacting this phosphate chain down here with this OH group from this nucleotide. So remember, this is actually RNA right here because this is the second OH right here. So this OH right here that you're adding, this attack is going to happen and be facilitated not only by the residues of the polymerase, the RNA polymerase right here that have this negative charge. This negative charge is going to bind the magnesium positive charge, which is going to facilitate this attack right here. This magnesium being held by this negative charge of the active site is going to basically keep these negative phosphates basically happy. It's going to keep them in the right spot so that this attack can happen. Okay. This didn't come out very well. So ligase is something that just seals basically the cracks left over by Okazaki fragments in DNA. It basically just joins and causes an attack from this OH right to here, from this other DNA to this other phosphate. This is a video usually. Sorry, I'll post this. But yeah, this hydroxyl group is going to be brought together with this adenosine phosphate right here to seal the full DNA crack. And you can tell that this active site right here is going to facilitate that. You can pause right here if you want. I'll post this. I'll have this PDF posted, but this is like the full reaction. If you want to see the details and everything, you can see all of basically the OH right here like we just saw, and it's going to make that attack with the phosphates here. Phosphates will join, and you can link the DNA, and you'll be okay. Awesome. Another cool way to use enzymes is that DNA helicases, so say that you need to unwind DNA, let's say you need to, make RNA from this, or you need to replicate DNA. So in this case with helicases or helicases, basically all they do is like spin in a circle, and they are just unwinding DNA the entire time by making contact right here and spinning those hydrogen bonds open. And remember, we talked about how important it is to make sure that why these are weak hydrogen bonds. It's so that you can have an enzyme quickly unzip something like DNA, because you need to access that information constantly. This is just super cool. This is helicase's quaternary structure. See how there's 1, 2, 3 of each of these isoforms. And in this case, it has a little RNA piece of it that allows basically the DNA to wind through. Unwinding recruits the RPAs. So the RPAs were those little DNA binding proteins that we saw. So basically when you first start unwinding, 2 helicases bind up and they start going this way, and all the unbound DNA has to get bound by these RPAs right here, because you don't want to expose nucleic acid, because that's super dangerous. So these both spin this way, and this is the slide that does a really good job showing that helicase is an enzyme. It's just super goofy the way it works. Basically, each of these little double pairs of colored subunits takes a turn binding the next set of DNA, and it just goes in this nice circle as it unwinds the DNA, as you can tell, getting more and more unwound. So it is kind of like a motor, like this says, a lot like ATP synthase, where it just basically keeps feeding on the reactions that it's made to happen. So this is another just interesting example. Then we'll get into some regulation steps, and then we should be done. This is a holiday junction. So all the nice stuff where it's like, oh, the enzymes crossed over and everything's fun, and this is easy meiosis. This is actually what's happening. Is this incredible interchange of paternal and maternal DNA. These are nightmares to learn about, though. So just for right here, this is just fun to look at how many proteins are at work making this happen so that you have variability in your offspring. Awesome. Okay. So let's end on some more critical stuff that we'll get into later. So we haven't actually introduced histones. All histones are these big positive, basically circles of proteins, and they serve as a way to basically wrap DNA around them. So remember, DNA is negative, and so they're going to wrap nicely around a positive histone, like that. If I had a whiteboard, we could do this a little better. What acetylation does is it takes the lysines, which primarily make up this big positive protein core, because lysines are super positive. What it does is actually add this little acetylation group here, and it negates that positive charge, and thus, instead of tightly wound DNA, now you have like much looser, like less tightly wound DNA, and now it's accessible. I really wish it wouldn't form those stupid structures. I don't know how to do that. Okay. So basically this loosens DNA from the histone grip and allows genes to be basically made from it, and genes to be turned on from it. So this is an activation step in most cases when you see it applied to histones. And this will get more as we get into like chapter 10. Methylation we kind of more saw on DNA as a silencer. When you methylate a protein or a residue like glutamine or glutamic acid, it's not always going to be a silencer when it's on protein. So we do silence DNA with methylation because it prevents promoters and stuff. And like we saw, it can prevent viruses from activating that are in your DNA. But on a protein, and as we'll see in histones, sometimes this can actually serve as an activating function. Like it can attract like another activator protein. Sometimes it attracts a repressor protein, though, so it's kind of variable in that case, especially on histones. Okay, phosphorylation is a great one. As we talked about, this is like a very key on or off switch for proteins, because if you add this big charged and large group, the proteins will usually undergo a conformational change of the 3D structure, because you've added to one of these amino acids this big disturbing structure right here, and that can shift the entire 3D shape of the protein even though you didn't change the primary sequence of the amino acids. So this is a very, very key concept to have in mind when you see proteins basically turn on. And we'll see this when we cover cell signaling in like brain chemistry. This is the last critical modification, and then we'll get into some medicine, and then we're done. This is ubiquitination. What ubiquitination does is it signals for a protein to be tossed in the trash and degraded. Okay, this is how you get rid of proteins after their life cycle has come to an end. Ubiquitinases will add this little chemical group here, this U, to a lysine. This is a signal for your cells. It's a pretty universal signal for degradation, and this enzyme is going to basically signal it for consumption and recycling of the amino acids. This is how you get rid of proteins. And like we talked about, we don't have a protein that can actually degrade, bad proteins like tau or prions. We do have something just in the last like 5 to 10 years though, PROTACs. Proteolysis mimicking chimeric molecules. I know that's a mess. They actually make this process happen to specific proteins that we want to destroy. We've never had that power before. So let's take a look at that. So we have a lot of diseases out there. I'm a cancer biologist, so we have to attack this disease from all kinds of steps. In some cases you have, DNA making genes that are very bad, and we want to get rid of them. Those genes turn into RNAs, and ultimately they turn into those functional proteins. At all 3 steps, we have things that we can actually send in to destroy or silence or modify the genetic material in each of these 3 cases. With DNA, we can send in a targeted CRISPR enzyme and nick, basically a bad part of a gene and change it, maybe, right? With RNA, we can send in synthetic silencing RNA interference. So these are these complementary microRNAs. They go in and they cause it to get diced. PROTAC is our answer to the protein question. We never used to be able to get rid of proteins. Remember how I showed you that ibrutinib? It would just go in here and plug a protein, but it wouldn't necessarily destroy it. It was still around. A PROTAC causes it to get destroyed. What a PROTAC does, and it's right here, is this tiny linker between the E3 ligase, this is basically the trash designator, it's going to force a connection between the E3 ligase and whatever target that we tell it to. This is going to cause the protein to be degraded by the natural mechanisms of your cell. So this is super interesting, and this is still in its infancy when it comes to clinics, but this is a massive tool, and you probably won't hear about it the same way you do CRISPR, but it might be even more powerful. And this is what a PROTAC is. It is a synthetic link between a target protein and the E3 ligase that is responsible for tagging ubiquitin trash signals to proteins. The protein causes the link, E3 ligase attacks, as you see in this. It's ubiquitinated, it is targeted for degradation in the proteasome, and it's over. Okay? Now imagine if we could apply this to a prion, to an Alzheimer's protein, to a cancer protein. That's the power of a PROTAC. And PROTAC is something significant too, because we've been talking about how do we create a structure that accomplishes something for us. PROTAC is about as close as we have got to do that. It's still an organic molecule, but it's approaching that synthetic, amazing level. Now remember, this green region is attacking a specific protein, so changing this region to different targets is also a challenge, though, because this is very different. This is an emerging field. Okay, I think that's pretty much it for now. These are a couple other interesting modifications for proteins, dilation. It's fine. We don't really have a strict interpretation of what the adenine groups do. General stability seems to come up the most. Methylation is good for targeting proteins, so if you tag this to one of the amino acids, this usually attracts it to the plasma membrane, so it goes to the surface at least. I've used this in research a couple times to make sure that proteins go to the surface. It's fine, but it's not overly powerful. And ribosylation is even less well known. It has something maybe to do with the apoptosis signaling. So remember, apoptosis is the signal for cells to blow up. It may have some role that when these get ribosylated, the cell knows that something's wrong and so it blows up. At the same time though, we're not 100% sure what the other functions are. The way that cholera and diphtheria kill you, unfortunately, is they spit out a bunch of signals or enzymatic activators that cause this to happen throughout your cells, and too many of your cells die. So think about cholera when you're making water and whatnot and the disease is spreading. It's because that intestinal lining of yours is undergoing a ton of cell recycling, and it's really bad and gross, but it's a great way to spread back into the water system that people drink. So circle of life, right? This is just a beautiful picture of Scout, and we're talking about Merl phenotype. I don't know. So her melanin gene has different isoforms at different levels of expression. I don't know how this fits in with proteins. Just awesome. And we talked about, yeah, these are the last two slides. Sorry, this is huge. Protease enzymes, again, we talked about what these do. Essentially, proteases are enzyme degraders. So their active site is basically going to chew other amino acids off. So if you start with a typical large peptide and see how this will continue on, that's what this signal means. The aspartic acids of this active site will serve as a chemical facilitator to activate and attack specific regions in this bond and break this covalent bond here of this peptide, and you're going to be able to cleave it. This is how you get rid of proteins, because like you can imagine, you can't have the same proteins running around your cells all the time. Okay. One of the things that we do to ensure that maybe we're measuring DNA, we don't want any protein in our tube in lab, you can use an enzyme called proteinase K. It's pretty substantially universal that it's just going to degrade every amino acid it sees. So you can kind of silently think to yourself, like, that might be a very toxic chemical to come in contact with. So again, this is kind of the final area that we're at with medicine right now, or at least the mainstream, is that we have small molecules that can mimic the binding of the active site or mess with the 3D structure. That's an allosteric inhibitor, for example, that they mess with the 3D structure, not necessarily at the active site, but it screws up the 3D structure so bad that the protein unwinds or loses its 3D shape and it breaks down. These drugs are specifically designed in this case to only attack the proteins that the HIV virus relies on, not necessarily just your cells rely on. So in a good way, and I think these attack reverse transcriptase, I'm not 100% sure. These will cause damage to the HIV virus proteins that are needed for that, but they won't cause damage to your cells. So we are getting very good at targeting our therapies and targeting medicine, and that's where genomics is starting to play a bigger role than ever. All right, we'll pick up with wild DNA structures in the next